We are going to continue with our next country, which is Tunisia. I would like to ask if Professor Adnan Cherif is here with us. If not, then I would like to continue with Professor Suad Ruiz from the Laboratory of Biopesticides, the Center of Biotechnology of Sfax.
Thank you, Victoria, for this opportunity. I'm here, and welcome to the audience. So please, if we can start with my slides.
Yes.
So I'm Professor Suad Ruiz. I am the head of Laboratory of Biopesticide in the Center of Biotechnology of Sfax in the south of Tunisia. Our center is a center of excellence. Next slide, please. Well known at the international level and equipped with many facilities for Technologie organized in platforms, especially for analysis and fermentation at high scale. We have 6 research laboratories working on the different thematic like plant protection and improvement, like environment protection and development of solution, especially for industrial and health applications. Next slide, please. In our laboratory, we have a staff of about 20 researchers, between young and experienced researchers, in addition to the engineer and technician. Next slide. Next slide, please. Yeah, so as the title of our laboratory is focusing on biopesticide, and we are working on the problematic of pollution of the environment, of the land, and of the plants by the chemical pesticides. So we are developing new alternatives to replace this chemical pesticide by bio ones. Next slide, please. Next slide, please. As I said, the alternative is the biopesticides, and we have different research topic in our laboratories. The next one, please. Next slide. Yeah. So as I said, our main objective is to develop new solution to replace this chemical pesticide by bio ones, especially for agriculture and urban systems application. Next one, please. So we are looking for new strains, and then we try to work through the value chain to deliver to the market new solution related to the biopesticides. Next one, please. The origins are different. We are working on microbials and plants, and we are focusing insects, bacteria, and fungi. Next one, please. We have 3 main research programs. The first one is based on new bioinsecticide, especially based on Bacillus thuringiensis, and on plant-based biopesticides. Next slide. Next slide. The second one is the study and production and formulation of antibacterial agents. And the next one. Next slide is the study of or the development of protective agents against insect pests of vegetable crops. Next one. Next slide, please. Yeah, and as I said, we are working on the value chain to deliver our products to the market. through different projects. I will give a brief presentation of our collaborative projects in the frame of the funding Horizon 2020 and Horizon Europe. I will give an example for the first research project, which is products based on BT, Bacillus thuringiensis. Next slide. So the first one is a project, a collaborative project in the frame of Marie Curie RISE project. It's based on a value chain. Next slide, please. Value chain from the strain from the lab to the market, and in which we are working on the bioprocess, on the proof of concept, on the assays of the product on field, and then all the maturation, the economic maturation, and the delivery to the market. Next slide, please. We have also other little, this is just to show this project, which is in the profit of an industrial, which get a new line, production line for biopesticide in Tunisia. Next slide. We have other collaborative projects just to treat scientific problems related to the scale-up of the fermentation from the little fermenter to the 100 to 1,000 liters production. The next one, and our new project. Next slide, please. Yeah. Our new project, which is begin this month, is about the promotion of our products through the Mediterranean in the frame of a new project which is funded by Prima, and in which we have many collaboration with different countries around the Mediterranean Sea. And thank you for your attention. So my contact.
Yes.
Thank you.
Thank you so much, Professor Souad, for this presentation and for sharing all the opportunities and the different collaborative projects that you are already working on, and that you're open to receive international researchers from the different EURAXESS worldwide hubs to come and work with you on a specific project within the Marie Curie postdoctoral fellowship. So thank you so much. We are going to continue with, is Professor Habib Samouda here with us? I think I could see him online. Professor Habib, can you hear us? From the University of Sousse. I can see you are connected. I don't know if you can hear us. If not, then we can give you a little bit more time, and we will continue with Professor Kaloua Waldia. I apologize for my pronunciation.
Good morning, everyone.
Good morning.
I am Paula Waldia.
Wonderful. Thank you so much. That was that was much better than my pronunciation. Thank you.
No
I'm going to share your presentation with our scientists. So
thank you, Victoria, for your all efforts and kindness.
Of course, please go ahead. The floor is yours.
Thank you. Good morning, everyone. It's a pleasure to be here with you, and thank you for this opportunity. I am Paula Waldia, a professor at the National Institute of Research and Physicochemical Analysis. First of all, I would like to introduce my institute, the National Institute of Research and Physicochemical Analysis. It's located north of Tunisia and is a part of the Technopark of Sidi Thabit. Previous slide, please. It's a
Yes,
part of...
it goes on its own. I don't know why, but I will try to stop it so it doesn't go that fast. Yes.
It's not a problem. It's a part of the biotech pole of CD Thebet. This technopark is dedicated to biotechnologies, pharmaceutical and parapharmaceutical industry, and life sciences. Next slide, please. The National Institute of Research and Physicochemical Analysis, INRAP, is a public research institute placed under the authority of the Ministry of Higher Education and Scientific Research. INRAP is located at 30, previous slide, please, is located at 30 kilometers away from the capital, Tunis, and at 17 kilometers from the international airport, and it's near 3 industrial areas. INRAP is devoted to study complex analytical problems in different fields, to develop new analytic methods to meet national needs, and to valorize research results and promoting their exploitation by industries. INRAP is well equipped for the chemical characterization of biological active compounds, as well as the development of functional food products and packaging materials. Next slide, please. We have many collaborating partners around the world, from Africa, Europe, Asia, and America. Next slide, please. Here I will enumerate some projects related to our hosting opportunity, only project with relation to our hosting opportunity. And I eliminate 2 projects entitled Valorization of Industrial Fruit Byproducts, Development of Active Coatings to Extend Food Shelf Life and Reduce Food Losses. Next slide, please. Another Tunisian-German project with the acronym VPAC, Valorization of Olive and Olive Oil Byproducts for the Development of Active Packaging Materials to Extend Olive Oil Shelf Life. Next slide, please. A PRIMA project entitled Valorize Foods and Improve Competitiveness Through Emerging Technologies Applied to Food Byproducts within the Circular Economy Framework. Next slide, please. Now I will focus on our hosting opportunity. The objective of our hosting opportunity is to develop novel and biodegradable food packaging materials with enhanced functionality, able to extend the shelf life of many food products. Firstly, extraction of active packaging materials. Secondly, characterization of biopolymers and bioactive compounds. Thirdly, development of active biodegradable packaging. And validation of the active biodegradable packaging solution. Next slide, please. For the first step, extraction of active packaging materials, we will extract the potential applicant will extract biopolymers such as pectin, cellulose, lignin, mucilage, and antioxidant and antimicrobial agents such as phenolic compounds, hydrophobic fraction, following green extraction process. Then the objective is to identify and assess different industrial byproducts and waste materials, evaluate various green extraction techniques, optimize extraction parameters, and purify the recovered compounds. For the second step, characterization of biopolymer and bioactive compounds, the objective is structural, physicochemical, and biological characterization, the evaluation of the activity, the antioxidant properties of the prepared extracts, their antimicrobial activity against various food pathogens and food spoilage microbes, and assess the safety of the extracts for human consumption using appropriate toxicological approach. Next slide, please. For the third step, development of active biodegradable packaging, we have interest in the encapsulation of the extract to ensure long-term stability, to characterize the barrier, mechanical, optical, and structural properties of the obtained materials at different storage time, and evaluate the biodegradability of the developed packaging materials. For the last step, validation of the active biodegradable packaging solution, we will apply the active packaging films or coating on different food products and investigate quality, storage, stability, and shelf life improvement of these treated products. Next slide, please. And the last one, Thank you for your attention and good luck to all applicants. And if you have any question, please don't hesitate to contact us.
Thank you so much, Professor Kalua, for this presentation. That was perfectly 5 minutes. I congratulate, because sometimes it's a bit of a difficult task to explain all the different opportunities that the different universities, the research institutions have for our researchers. So this is definitely a master task and a master class. So thank you again for being here with us this morning and for sharing this information. Hopefully many of the researchers will contact you so they can do several projects with you and with your laboratory. Thank you. We will follow our Tunisian cohort with I can see that Professor Habib is present. Hello, we cannot hear you. No, if you can turn on your microphone, please.
Good morning, Mrs.
Yes, now it works. Yes, perfect.
All the
Thank you.
people.
Professor Habib.
I am a Prof. Habib Samouda. I presented my laboratory of energy and materials from University of Sousse, donc at Higher School of Science and Technology of Hammam, Sousse. We have
Can you see the presentation? Hello? Professor? I'm showing the screen. I'm showing your presentation. Can you hear us?
Good morning.
Yes, I'm showing your presentation.
Okay.
So please go ahead.
Okay. Alors, dans our laboratory research have 4 projects, 4 projects concord with training at our school, SSTHS. We have 3 formation on fundamental licence physics and materials and energy, and the PhD engineering of physics, and master of degree on materials and engineering and building. The next. Alors, our laboratory is from 2012. From we have progress with different time. We have submit last assessment by CNEAR in April 2019.
Can you see your slides, Professor?
Yes, yes. Various LabEM projected from our laboratory is 4, and we have Excuse me, I have not can terminate this exposition.
I think there might be some technical problems with Professor Habib, but you are going to get this presentation, of course, as well.
Yeah, كان ما بيجاني ربي.
So we have about 1 minute left. I can maybe go through some of the slides. Thank you so much, Professor, and I'm sorry that you had some technical issues. It doesn't matter because, of course, we are going to share your presentation with everyone present, and they are going to find out more about your laboratory at the University of Sousse and all the opportunities that are offered to our researchers. So we are going to continue with our last presentation by Dr. Yasser Hafani, who should be here with us. If you can confirm, Dr. Yasser.
Good morning, I'm Dr. Yasser Hafani from University of Manouba in Tunisia. I will be presenting my project idea, AfriOnc, entitled Africa Breast Oncology, based on the discovery of novel susceptibility and treatment response. My organization, University of Manouba, is a public academic institution in Tunisia. I'm affiliated to the Higher Institute of Biotechnology, Sidi Thabit, which is located in a great active environment in research and innovation, which is the Biotech Pole. Sidi Thabit is the first largest biotech institution in Tunisia and carries out research in biology, biomedicine, nutrition, and food science. My institution has the necessary research condition for this project and do have experience with international projects. We harbor Erasmus Plus, international career mobilities. We harbor also a key action to capacity building project Erasmus Plus, and I am co-leader of the Biotech Tunisia project. Also, I am a coordinator of the bilateral project of the Tunisian South African project, R&D joint grant, regarding the genomic and pharmacogenomic of breast cancer in Tunisia and South Africa. I am also a partner and member of the consortium of artisan food, which is the EU Research Innovation Program, PRIMA program. And we do have in my institution experience with the 7th Framework Program project, and also we have 2 projects completed and another project in Horizon 2020 already completed. AfriOnc project idea will be divided into the following two main objectives. First of all, the discovery with high throughput sequencing the genomic and pharmacogenomic variant of African breast cancer patients. Currently, we do have research innovation project discovered novel variants of the most common genes of breast cancer with level 2 of innovation, TRL2. Second main objective is the discovery of molecular broad, the discovery of novel compound through molecular broad screening and pharmacogenomic evaluation of novel natural bioactive compounds based on cell assay for cancer. This agent may be involved in pharmaceutical industry, which consider the essential scope worldwide overcome of breast cancer to minimize the high cost of anti-cancer drugs. My institution is very active in terms of clinical research. We access to clinical samples, a biobank and collection of oncology patient samples, as well as clinical information. We access to facilities and technologies such as animal house, genotyping, cell culture technology, qPCR platform. We do have experience participating in international research innovation projects in different type of tumor, having links with several public and private hospitals, research centers in Africa that are already collaborating with us. And our background and experience provide a good opportunity for participating in multidisciplinary research projects. I am co-founding member of African Pharmacogenomic Consortium. That was in 2018, and I am very active in organizing seminars, international seminars and symposium like the one done in Cape Town in 2023. Our organization and myself, I do have specific skills and technology expertise to the project in terms of SOP, biorepository, oncogenomic data collection, cell-based assay, data-driven insight in cancer care, modeling human disease, metagenesis and phenotyping, gene chip technology, artificial intelligence and machine learning expertise. Thank you so much for your attention. And this is my contact. I would be happy to work with you and follow up this project with you at the world-class level. Thank you so much, and thank you for your attention.
Thank you so much, Dr. Yasser, for your presentation. And with that, we have finalized another country, which was Tunisia. So thank you to all our presenters presenting the different laboratories, institutions, and universities of Tunisia, to our researchers that are connected here today.